Hello, everyone, and welcome to today's webinar, Interim Analyses to Optimize Late-Phase Clinical Trial Decision-Making. We are excited that you could join us. My name is Laura Beth Ezel, and I'll be moderating today's event. Now, today we'll be doing a deep dive into what interim analyses are, as well as some design and operational factors to think about as you incorporate them into your development plans. Today, we are thrilled to be joined by three speakers. We have Brett Gordon. He's the Associate Vice President, Product Delivery at Rowe. Ben Vaughn. He's Chief Strategist, Biostatistics and Protocol Design at Rowe. And Scott Mollan, Associate Director, Biostatistics at Rowe. Before we get started, I have just a few housekeeping items. Please note the slides will advance automatically throughout the presentation. To enlarge the slides, click the Enlarge Slides button located in the top right corner of your presentation window. Now, if you need technical assistance, click on the Help widget located on the bottom left corner of your console. Now, we encourage you to submit questions at any time throughout the presentation using the Q&A widget that's at the bottom of your console. Now, we will try to answer these during the webcast, but if a fuller answer is needed or we just run out of time, it will be answered later via email. So please know that we do capture all your questions, and you're welcome to go ahead and submit those as you think of them. Well, to kick off, I'd like to ask each of our speakers to give a quick introduction of themselves and their work. So Brett, would you like to go first here?
Yeah, thank you very much. So I'm Brett Gordon, Associate Vice President of Project Delivery at Rowe. I've been in clinical development for about 25 years, the past 10 at Rowe, where I'm responsible for both operations strategy and client oversight. I spent much of my career helping biotech clients across a wide variety of therapeutic indications, but with an emphasis on neuroscience and analgesia development programs, many of which are increasingly building in interim analyses to better guide decision-making. I'm happy to be a part of today's discussion and lend an operational perspective on IA planning and execution. Thank you.
Welcome. Ben, tell us about yourself.
Hi, Ben Vaughn. I'm a statistician. I've been in industry for 22 years, much of that in late stage and marketing applications. And I've supported a very large number of these trials that do use these interim analyses, particularly for adequate and well-controlled trials that would be used for a marketing application. Looking forward to the discussion today. I hate slides and love questions, so please feel free to drop those in the chat.
Absolutely. Welcome to you as well. And Scott.
Hi, I'm Scott Mollan. I've been in the industry a little over 17 years doing biostatistics. A breadth of my experience is in medical devices and orthopedics, pain studies, mostly in the late phase space, similar to Ben. Had a lot of interim analyses in my day, and looking forward to sharing the knowledge with everyone out there. Thanks.
Absolutely. Well, thank you all. So let's get right to it here. So I'd love to start at the highest level here. What is an interim analysis, Ben?
An interim analysis is any look at the data while it's ongoing. These can be unblinded or blinded and related to efficacy or safety. For the purposes of our talk today, we're really focused on those that are looking at efficacy and could be used in adequate and well-controlled trials. We want to focus on methodology that allows you to use your trial for a marketing application and still say that you have tight alpha control, while still having the opportunity to look at your results and adapt.
Okay, and so let's get into some specifics here. You know, what are the different types of IAs and what's the benefit of each? And what are the risk or penalties of each? Ben, we'll start with you.
Sure. So broadly, there are three types of these interim analyses that we're focused on today. As mentioned, there are safety analyses, there are early phase approaches that you can use to trim down treatment arms, where you start with a bunch of doses and end up with one or two at the end with a greater sample size in those. But today we're going to focus on the ones that you can use in adequate and well-controlled trials. And there may be some others that I'm not mentioning here that are less commonly used, but we're going to focus on stops for efficacy, stops for futility, and sample size adjustments. So with a stop for efficacy, you really are focused on winning fast. You want to be able to take a look midway through the trial and see if you've hit a very high bar p-value. If that's happened, then you can stop the trial and declare success. The risk with this is that you may not get full information on everything else. It really is focused around a single outcome. So if you stop the trial early, you may not be able to have statements around the statistical significance of your secondary endpoints. You won't get as many drug exposures, and you won't get as much safety information. On top of that, if you fail to hit that high bar, you have less alpha at the end of the study. That is that you have to hit a smaller p-value at the end of the study versus the most common 0.05 p-value. With a stop for futility, it's the same idea. If the drug just isn't working or your trial is designed poorly, you take a look at the data partway through, and if it looks particularly bad, you halt the trial and declare it futile. The risk there is that when you do this, sometimes the second half of the study turns around and you've stopped a trial that would have ultimately been successful. It's hard to say for sure because it's been halted, but occasionally you will have trials that get a read of futility that's non-binding, the trial continues, and ends up having significant or close to significant results. And had they halted it, that would have been a beta error or a case where you had a false negative or a false signal to stop the trial. And then finally, you have sample size adjustments. And there are a few different types. With the efficacy and futility stops that I just mentioned, each of those require an unblinded independent committee that has no other interaction with the study participants, sites, staff. They're completely independent. Likewise, you can have an unblinded committee that looks at the data to date and decides if the trial is on track. If it's looking great, perfect. You stay the course and complete the trial with the planned sample size. If it's a little off of what's planned, you can increase the sample size. This increase is capped at up to 50% of the original sample size. This is not a math thing. It's more of, I think FDA just gets uncomfortable if you start increasing the sample size that much. That would say that something was wrong with your original design. Additionally, if you do this increase halfway through, that would mean that the second half of the study was twice as big as the first half. Additionally, you can do a blinded variance re-estimation where you just look at the variance thus far in the trial. The variance can drive the required sample size. And so if there's a signal that perhaps your variance is larger than expected, then you may want to increase your sample size. There are some risks with that approach that we can talk about a little bit more later. And then finally, there are external information updates. And with that, perhaps you have another trial running and it locks and you have new information that you didn't have previously. If this is done in an entirely blinded manner, you can increase the sample size based on that external information with no penalty at all. Back to the unblinded committee, there is a small penalty with that, but it's not an alpha spend. You don't change the p-value that's your threshold, your 0.05, when you're doing the final look at the data. However, there is a slight adjustment to that p-value and how it's calculated, so that would build in a very, very small penalty.
Just to chime in on the blinded variance re-estimation, it's been alluded to, it is something that can be done, and you can adjust based on seeing a wider variance than originally assumed. In practice, this type of sample size re-estimation typically isn't advised. Knowing how precise your estimates are with regards to variance is always a nice thing, but it doesn't really tell you anything about the estimates themselves for the treatment effect and how far apart they are. And you might have a very large variance, but if the treatment is performing very well, then you may be able to overcome that increased variance with no additional sample size. Conversely, you may have really good variance estimates, but if the treatment and, say, the placebo are performing identically, there won't be a sample size that you could possibly come up with that would be feasible to see a difference. And so, the amount of situations where you would want to consider such a method are rather limited in scope.
So I'm seeing a few questions come up in the chat, so I'll address those really quickly. The first one was how to decide the true variance, and I think this may be related to the blinded variance re-estimation. And the problem there is that because it's blinded, what you do is you take the pooled variance. You're not looking at individual treatments, you're looking at just the overall variance. And you assume a fixed difference between the two treatment groups. And usually this is the one that was in your original power calculation. And based on that, you can do your math and see if those the variance for the treatment difference back calculating that is bigger or smaller than what you expected. The problem there should be pretty obvious right off, though, and why we don't recommend these, which is, let's say your treatment is doing a lot better than you expected. All of a sudden, this pooled sample is a lot wider, and the pooled variance will be a lot bigger, which would suggest that you need a bigger sample size, when in fact, your treatment is just doing a lot better. Conversely, if they're a lot closer together, and this is what Scott was mentioning, you would have a great looking pooled variance, but if you were wrong about that original treatment difference, all of a sudden you're getting this read that things are looking great or better than expected. You think you don't need to increase the sample size, and all of a sudden you're doing the wrong thing. So we don't recommend this unless you have a great deal of external information, a 505b2 on a drug class that has been very well studied, and that treatment difference is well established, well known, and really you are just looking for the variance piece in your particular study. Otherwise, I would avoid it. And I see a second question, is there an alpha cost with the futility interim analysis? And the answer is no. The agency is actually perfectly fine with you doing these with pretty much no penalty. The penalty is implicit because what you're doing is you're giving yourself another chance to fail. You're not giving yourself another chance to succeed. Alpha is when you declare success and incorrectly. Where you have two drugs that are the same, and every now and then you get lucky and they separate, and you declare that as a difference when it's not truly a difference. Beta is the opposite of that, when you have a drug that works where it should separate and you don't find it. And so there's a possibility in a futility analysis to declare failure when, in truth, the drugs really need to separate from placebo. So there's no alpha cost. You just need to be careful because there's a little bit of a power hit. And when you're looking at the data, let's say halfway through the study, there's variability and it could look a little worse. And then in the second half of the study, it does a little better. And if you cross that threshold where you declare futility, well, there's no undoing that. You've declared futility and study's over.
Just to piggyback off that, with respect to the futility analysis, Ben's 100% right that formally there is no alpha cost, but I will see some sponsors, take a slightly more conservative route whilst to guard against people saying, well, you looked at the efficacy data, theoretically you could have made a decision off of it for efficacy. And those types of situations, if you're wanting to guard against that, you would typically take a very small alpha hit. We're talking 0.000001 sort of thing, just to show that you're addressing those concerns of maybe you could have made a decision. But formally speaking, you wouldn't need to take an alpha hit.
Yes, and we're happy to drop some references. Another approach would be to use the exact same methodology that you would use for the sample size adjustment, where you're pretty much taking a weighted average of the two halves of the study. And with no sample size increase in expectation, there's no hit to your estimate or your p-value with that, assuming that the first and second half look about the same. So there are a few approaches to be conservative, make sure that everything feels like it's been addressed. And moving on to something else within that topic, I see a question: do you need FDA approval for all types of interim analyses? Mostly, yes. And we'll go through each one of them. For an efficacy stop, absolutely yes. You must spell this out. It must be in the protocol. You need to have your charter prepared, which describes how you will unblind the committee, what communications the committee can have with people who make decisions in the trial. Every possible base must be covered with that. They're going to want the full SAP describing how you're going to do your alpha spend. If you have a sample size readjustment in there, they're going to want to see all the formulas. Just everything must be spelled out perfectly for that efficacy stop. And the agency is a little suspicious of those. Can you do them? Absolutely. Can you do them in adequate and well-controlled trials? Again, absolutely. But you need to get the agency on board and get them convinced that you have checked all the boxes. Otherwise, you're just going to get information request after information request. So some caution with those. For futility, same thing. Let them know you're not going to get that many questions. The main thing is that they just want it to be a non-binding futility stop. Won't get into the math of that, but basically, they don't want you to be forced by what you get from that that interim read. And so with a non-binding futility stop, you will get the advice that it looks like the trial is not on track, and then you just make the decision whether or not to adjust based on that. For unblinded committee for sample size readjustment, same thing as with the efficacy stop. You really need to spell everything out, describe what will drive the decision for increasing the sample size, how many blocks of subjects you will add. You don't have to go from just, no increase to increase by 50%. You can do it by increments of, say, 10 subjects, 5 in each group. But that needs to be spelled out, and you need to get agency buy-in on that. For the blinded variance re-estimation, because it's blinded, you could, but again, this is something that we see done, and we just do not recommend doing that. But you probably would not need a ton of agency buy-in on that. You would probably need to notify them that you're increasing the sample size. Likewise, with external information updates, I would say notify the agency that you're increasing the sample size, but you probably will not have extensive interactions with that, with the exception of if they feel like the sample size that you're going up to would give you a significant p-value for something that is not clinically significant. If you have a big enough sample size and just a tiny separation between active and placebo, it'll come up stat sig because you have such a large sample size and your estimates are so tight around those means. And so if you start getting into the range where you could declare success on p-value and just do not have a significant, or a clinically significant separation, then you're going to get some questions. Also, you can't lose sight of good clinical practice. These are experimental drugs, and if you are exposing a very large number of people with a potentially small benefit, then it might become an IRB issue.
Yeah, excellent. Sounds from what you've shared, though, it's not just about cost when you're evaluating the benefits or risks of these different options. So what are some other perhaps underestimated factors that should be considered here, Ben?
Well, there's, we talk about statistical cost, but there's also dollar cost. There's uncertainty. You're starting a trial not knowing how big your sample size might be. You may have to suddenly stop the trial for efficacy, stop the trial for futility. This is something that your sites probably are not going to like. Or you could show up to your sites and say, hey, we need to recruit 50% more people. So there are risks in that. Your estimates of when you will be finished just go completely out the window when you all of a sudden have to add up to 50% of your sample size over again. However, this is not necessarily a linear cost. Increasing your sample size by that much wouldn't necessarily add 50% to the cost. Database build, same cost. Statistics, same cost. However, there are also costs associated with running these analyses. You have to do a database freeze of some sort, make sure all your data are clean to your satisfaction. We can return to that. And you have to actually run the analyses. If you're going to have an independent committee, you have to schedule some very busy people and get that time and get the read back, and then adjust accordingly.
Yeah, and how many IAs can be or should be conducted, Scott?
So, I mean, beauty is in the eye of the beholder there. And it often comes down to the logistics. Theoretically, you can have a very large number of them. You could look every day if you wanted and were willing to take the hit. But practically speaking, that doesn't make sense given the logistical hurdles. Commonly, I will see anywhere between 1 and 3, maybe spread out throughout the course of the trial. And that is really field specific. In the device realm, I have seen 3 to 4 on the regular, but in a drug development setting, you're often looking at typically only 1 interim analysis.
I would agree with that. I don't think I've ever seen one in a pivotal more than one in a pivotal trial. It can be done, the math supports it, but when you start looking at the data that much, the math gets a lot more complicated for the adjustments that you need to make. And additionally, it's hard to support the idea that you are not just tweaking the trial and it feels a little too hands-on, and that you really are not keeping it adequate and well controlled. And so I would caution against doing more than one. The agency is perfectly fine with 1 interim analysis. Starting to get into more than that, I would be concerned. And actually, I should clarify, that's speaking strictly of CDER and CBER. In the device realm, there's a lot more wiggle room, and they're a lot more open to these things.
Yeah, we're going to get to the device realm here in a little bit. But at what point in a study should an IA be done, Ben?
I really like 50%. And part of that is if you're ever trying to guess at when you should do anything in statistics, 50% is a great guess. This is pretty much how I got through grad school. If you know the answer, you can work out the math backwards. And same thing here with the 50% cut. A lot of the things simplify in terms of the weighted averages you might do, how you calculate the variance. And so it's got a lot of good properties. And beyond that, just in terms of the balance of information, you've got half the trial done, and that is a good place to get a somewhat stable estimate for what may happen in the second half. Now, when we're doing a sample size readjustment, we call it conditional power. And what we're doing is we're conditioning on what we have seen thus far. That's locked down. That won't change. And then we have a second half of the study, second cohort of the study, let's not say half, because the question is, when should you do it? You have a second cohort of data that's still variable. And so the first half locked down won't change, but then we have to make assumptions about the second half. Is that second half going to be identical to the first? Well, then we're putting something kind of variable in there, because that first half estimate we just did, it's not perfect. It's not a known constant. It's just a guess at what's happening. And so we can use that for the second half. We condition on that. We say first half locked down, we know it, and we're going to assume the second half looks like that. But you can also use your original power calculation. If you had another trial lock, you could use what you saw in that second trial. Presumably it went to full sample size, and you could use that estimate for your assumption of what happens in the second half. You could do an average of what you saw in the first half in this other hypothetical locked trial. It's whatever you want to put in there. Most commonly, though, you use what you saw in the first half. Those are your sites. That's your exact trial. It's how it's being conducted. That's probably your best guess of how the second half will work. And so with that, the 50% mark is a pretty good balance of having good information while still having enough outstanding subjects to adjust.
Yeah, and then, to that point, with number of outstanding subjects, to consider here from an operational perspective is, 50%, I think it's a really simple marker, right? Like Ben was just explaining, for when you would want to do a data cut. But one thing that you need to consider is where you are with overall enrollment of your trial at that point. Because it's conceivably possible that once the 50th percentile, once that patient has actually reached that primary endpoint and you would have the cutoff for your interim analysis, your trial may be fully enrolled at that point. And if you're going to do, for example, sample size re-estimation that might increase the sample size, you need to consider that there may be a pause in enrollment, and then you're going to lose momentum with enrolling patients in that study. I think one thing you definitely need to consider is looking at your planned enrollment trajectory at the beginning of the trial to make sure that it aligns with the data cutoff point that you're choosing for your interim analysis.
Yeah, and to be clear, you do not have to pause enrollment unless you've hit full enrollment. And you also don't have to pause enrollment even if you have hit full enrollment. You just need to be aware that you're running a little past that. And that's allowable. The agency is not going to get completely bent out of shape if it turns out that you're not going to increase the sample size and you ran a little past your planned enrollment. If you go a lot past your planned enrollment, that's when they get a little more concerned about what's going on there. But typically we can run these with no pause, and really the cost at the middle of that trial conduct is in terms of whatever data cleaning and freeze you want to do, and actually creating the displays, analyses, and the committee costs.
Right. Yeah. And one other point that I'll also make, Ben, is that, again, if you ultimately do need to increase your sample size to sample size re-estimation, one thing that you'll want to consider and start these conversations very early on, of course, are, do you potentially need to have additional sites at the ready to contribute to enrollment? Especially if it's a rarer disease population where by the time you've now hit 50% or even fully enrolled your study, maybe your original sites are capped out on what they're able to contribute to the overall enrollment of the study. So again, there may be additional cost implications, as Dan had laid out really early on in the webinar this afternoon, that you need to consider, again, when thinking about, do you need to add sites to contribute to those additional patients that you might need to enroll based off your sample size estimation.
So I see another great question here in the chat. Can the IA be done based on country-specific or specific countries before expanding to other countries? And that partially gets the operational considerations that Brett just mentioned. There could be some challenges with that. But the other part of that is, it's kind of you can do anything, but is it a good idea? And that goes back to what I just mentioned, where and I assume this is for a sample size adjustment where you'd look at that and then continue. The concern would be that you're assuming the second half will look like the first half, typically with that. And so if you're operating in specific countries and then you expand to other countries, which may have different standard of care or cultural expectations around a disease indication or all kinds of things. I work in the pain space a lot, and there's some regional variability in pain reporting. And so the risk there is just that that first half doesn't look like the second half. And I frequently say your interim is never wrong. It's just maybe not what you expected it to be. The numbers are what they are, but if they are not a good estimate of what you're trying to do, what you're trying to assume about the second half, then you're taking on some risk there. But could you set up something like this? Absolutely. Brett, can you think of any operational challenges?
Operational challenges, yeah. I mean, so there's a number of them in general. I think with the IA, and I know we're going to get to this probably in a later slide, so I don't necessarily want to go through them all right now. But definite things that we'll need to consider, from the early outset are everything from factoring in how you're going to work through project plans, putting the information about your interim analysis, for example, in your clinical monitoring plan to make sure that we are actually adequately planning for what data are going to need to be monitored to support the IA. And again, I think we're going to talk about this in a little bit more detail later on, but then I wasn't sure if you were specifically pointing to anything else.
Yeah, good points. And so let's talk about, who sees the data then at this point. Who is unblinded for all these interim analyses, Ben?
Okay, so pretty much it is either blinded, which means that you're not actually looking at, unblinded treatment codes. You don't know the specific results of the trial, or you have to have a committee that is independent and has no decision-making capabilities in the day-to-day operations of the study. And occasionally you will see where a sponsor representative has a little more insight to the data. However, that's risky, the agency doesn't like it, and they really need to be firewalled off. It needs to be somebody that has nothing to do with the study otherwise. But typically you're going to have an external committee with the usual financial disclosure that they have no financial interest in the study involved. And you can also do this as a purely statistical review committee. So one option is to get some outside experts, doctors, clinical trialists that you really trust to make these decisions. You want a statistician on there to look at the results of this and make a judgment call. These should largely be driven by the math, but occasionally you may want those external experts to look at some other things. You may be looking great on that primary, but based on their expert decision making, they may see that your secondaries are looking a little weak. And so despite the fact that you are seeing a very strong efficacy signal on the primary, that may say, hey, do an efficacy stop. They may recommend that you continue the trial just because there are other things going on deeper in the data. However, this would need to be spelled out in great detail what their mandate is in that charter. The other option is that you have a very tight charter where it's just somebody looks at the number, runs their finger down the table and says, increase by this much. Or if you hit the threshold, stop the trial for futility. If you hit the other threshold, stop the trial for efficacy. So when you have a purely objective read like that, sure, you can get a committee of experts, or you can just have a statistical review committee where you have two statisticians, they check each other, check the numbers, make sure everything looks great, and deliver that recommendation. But again, for anything unblinded, it needs to be an independent committee that is not otherwise involved in the trial.
Just to add, earlier Ben mentioned firewall. From an operational perspective, it's super important that the committee, the statisticians, whoever it is, be firewalled from the rest of the study team. And furthermore, you would want to have somebody at the sponsor who is also firewalled, someone that you're giving the recommendation to, who has no role in the study other than to look and accept or reject the recommendation, so that they're not influencing future day-to-day conduct of the study. And you'd want to make sure that person's senior enough to have the capability or the responsibility of making that decision given the obvious financial impact to the organization.
Yeah, great points there. And what does the FDA want to see before they agree to these plans, Ben?
I think we covered some of that earlier. You really need to, if you do have an independent committee and you're doing unblinded look, you need that charter, which describes, the firewall that Scott was just talking about, what reads come back. Is it a sample size adjustment alone? Do you have futility and efficacy built into that as well? You can do all three. If that wasn't clear from what we were saying already, all of those can be built into a single protocol. But they're going to really want to have that spelled out in explicit detail. The agency will also want to see this in the protocol. It needs to be described there. Usually you just can't have this separate charter document. The protocol needs to acknowledge that you could potentially have this stop. Likewise, this is less of an FDA issue, But for your IRB, the informed consent also needs to address this, particularly for futility and efficacy stops. You may have subjects that are ongoing in the trial, and you need to spell out what's going to happen with those subjects if you suddenly stop the trial. And then finally, you can put enough detail into the protocol to cover this, but typically the SAP needs to be fully fleshed out, describing whatever adjustments that you will do. So in the case of an efficacy stop, you need to do that alpha spend calculation. You need to put in the charter how much alpha you will use, what p-value do you have to hit to stop for efficacy, and then how much alpha you have left for the remainder of the trial. Additionally, for sample size readjustment, you need to describe how you will combine the data from the first cohort and the second cohort, or if you do multiple cohorts, how all those will be combined, and what variance formulas you'll use for that. So everything really needs to be spelled out in explicit detail to get the agency on board and agree to this. However, they generally do. They may have a few questions, or they may pull out a few threads there, but if you come to them with sufficient information, these are allowable methods for adequate and well-controlled trials.
Yeah, thank you for going through that, Ben. And so we've obviously talked mostly about drugs here, but I do want to give any device friends some acknowledgement too. We touched on this. So you've already alluded to some of the key differences between biopharma and devices, in that the FDA may be more amenable to seeing multiple IAs for devices. So are there other differences to consider, Scott?
Sure. So, generally speaking, anything that works in the arena of drugs is going to be considered acceptable for devices. But, as we mentioned, in the device realm, sometimes you can do a few more interim analyses. If you want to have one early for, say, futility, and then a later one for stopping for efficacy or sample size reassessment, you could do that. Or that sort of thing really isn't on the table for drugs. Likewise with drug trials, your data is supposed to follow CDISC requirements, but in devices, it doesn't need to. So there's a bunch of nuances in how you work on a device trial that you would still be rigorous, but perhaps not as rigorous as in the drug space.
Yeah, and with that, I would say come up with something that aligns with what drug division would consider acceptable, and then stretch the boundaries a little bit in your favor and see what device division says. I have been shocked at what I've gotten away with, and so that's certainly again nothing unethical, but stretching the boundaries a little bit, and that creativity can certainly help you in, for example, getting a device on market with an initial claim and then following up with new claims and new indications.
Great. And so just switching gears a bit to talk more about operational implications here. What do you do differently in operations for study startup for trials with these designs? Brett?
Yeah, excellent question. So first of all, I think taking a step back, I think most likely everybody is familiar with the phrase, beginning with the end in mind. When you're planning for an interim analysis, it's more like begin with 50% in mind. So at the early outset of the study, you want to make sure that, again, you're building into your project timeline, you're building into your project plan all of the necessary steps operationally to complete your IA. Ben's already alluded to the analysis plan and making sure that that clearly defines all of your interim analysis procedures. But then from an operational perspective, again, and I touched on this a little bit earlier, you're going to want to define, for example, in your clinical monitoring plan, what you're expecting needs to be monitored to actually, pull off your IA. Thinking about that a little bit more, an interim analysis is not simply your final database lock carried forward. It can be both simpler and more complex at the same time. You don't necessarily need to SDV, for example, 100% of all of your data points. It's going to largely be dependent on what you're looking at in that IA, which typically is going to be your primary and or key secondary endpoint. So a lot of times what we'll do is advise sponsors to limit data cleaning to only the essential data that they actually have to monitor at that point, and not necessarily spending their time SDVing all available data. A few other things that you need to consider when planning for an IA as well is that, again, when I said it's not simply your database lock carried forward, when you get to database lock at the very end of the study, that's the sole thing you're focused on. Sites have already moved beyond enrollment. The last patients are completing treatment and maintenance period. Typically, when you're doing an IA, you're doing this in the midst of ongoing enrollment. You also need to be cognizant of the impact that this is going to have on sites, sending CRAs out there, cleaning data, asking them to enter data in a timely fashion. Again, thinking about that they're going to have to be continuing to enroll the study, deal with the operational challenges of enrollment at the same time that you're now asking them to plan for a data cutoff point to support your IA. There's a number of other considerations as well that we can go into, but I think that was at least an overview or starting point.
Now, one other consideration I'd like to chime in on is we talked earlier about firewalls. One of the things that the FDA is concerned of when you're doing an interim analysis is the potential for injecting bias into downstream events. And so to that point, when it comes to things like budgets, site communication and the like, it's really helpful if you can to communicate or plan for the trial in sort of the worst case scenario. And one example there would be, say we're doing a sample size reassessment and all of a sudden we need to add 100 more patients. It's going to need 5 more sites, what have you. Doing an interim analysis and then all of a sudden saying, oh no, we need a change order for X, Y, and Z, you're showing your hand to the sponsor, the study team, that is, and they'll realize, oh, things aren't working the way we wanted. Likewise, sites could think things aren't working the way we were hoping for, and that could potentially change how they recruit patients or maybe make other protocol related decisions. So planning for things up front negates that showing your hand, so to speak, and can allow for minimal bias to creep in.
Yeah, and just to clarify on 2 points there. First off, from the start, making it clear that actually a sample size adjustment means you're in striking distance. It actually generally means that things are looking good. In terms of the operating characteristics of this, if you've got a sample size adjustment that's within that 50%, probably means the study will succeed because you're able to add enough subjects to still have, say, that 90% chance of success that you had based on your original assumptions. So sample size adjustment, if it's not futility, all of a sudden this is a good read on most trials. So making sure that sites understand that, hey, looks like things are pretty much on track. We're just going to bring up the sample size a little bit to ensure that we hit that p-value is good news. So again, managing expectations there. And the second point is, Brett did talk about SDV and data cleaning and all that. Not recommended, but if you're willing to take the risk of some dirty data, it does not necessarily have to be done. You can do this on dirty data. If the data change for the final look, you use whatever the final data are. Just know that there is a risk in dirty data, and it has to be acceptable for what you're planning to do.
Yeah, and I think as a potential mitigation for that as well, and we're seeing this more and more in trials nowadays, especially in the neuroscience and analgesia areas that we have a large degree of expertise in. Most of the endpoint data is being collected by some version of direct data capture, whether it's ePro, eCOA, whatever else you want to put after it. data doesn't necessarily need to be SPV because it's direct entered again by either the clinician or the patient themselves. So what we will do is set up programs to centrally monitor that data, basically making sure that it's complete, looking for if there's any outliers so that we can kind of course correct as we go along if data look odd in terms of data entry. But again, that's data that we're monitoring centrally. And the more data that we're seeing that you're able to enter by direct data capture ultimately leads to less data that you need to go on site and monitor. So it's really less a question of dirty data. It's more how much data can we centrally monitor versus, again, sending CRAs on site to actually truly SPV.
Yeah. And what needs to be done is you just make a list of anything, any field that contributes to that primary endpoint, assuming it's just the primary endpoint that you're putting into this interim analysis, and check your boxes. Decide what could be dirty with this, and what or incomplete or problematic in another way. And to Brett's point, I love ePro because typically, very frequently, your primary endpoint is in an ePro, and so that may just leave one or two other things which are generally trustworthy. For example, if you're doing an analysis that takes into account the reason for dropout, then that would also need to be part of this cleaning process. But you can go through everyone who's not complete, pick out the ones that maybe you're not so sure about, and really hit the sights on those and really narrow down the problem. So yes, the ePro and direct data capture really simplifies this process a lot.
In one point of caution, just to throw it out there. So sorry, this is like with ePro and direct data capture. Sometimes that data is integrated within our EDC system. Sometimes it's third-party data, and just thinking a little bit more about third-party data, because this is definitely a place where people can get caught. You want to make sure that you're setting up your data transfer agreements early and actually testing your data transfers well in advance of your interim analysis, so that the first time you're getting data from, say, a central lab, a central ECG vendor, whoever it is, you're not running into reconciliation issues immediately at the time of your IA. So again, just like you would prior to a database lock, making sure that you're reconciling that data periodically along the way, just to avoid any hiccups with reconciliation.
Yeah, and if you stop for futility or efficacy, what do you do with everyone currently in the study, Brett?
Good question. Ben alluded to this earlier. You really need to think about this upfront in your planning. We've seen sponsors handle this different ways. So if you wind up stopping, depending on, I think, the complexity of your trial, one option, of course, is just discontinuing all patients as soon as possible. But you also need to consider, based off the drug that they're actually taking, do they actually continue taking the drug for a period of time, and then you eventually try, let's say, if the drug that they're titrated up on, do you titrate this back down? Do you continue to follow those patients, possibly for safety over a period of time, but no longer for efficacy, since you might be stopping your trial? Again, these are all things that we really look at on a case-by-case basis based off the design and complexity and specific drug product that's being given to patients for those trials. So Ben or Scott, I don't know if you have anything else to add there.
Yeah, consider an open label follow-up, safety follow-up, and just don't put subjects into that. That's a common approach with this, where you have those subjects, you recruited them, frequently you need more safety data, more exposures. And so that gives you an opportunity to not waste those efforts that you've put into recruitment and continue to get more data with these subjects that meet all your inclusion, exclusion, and might be hard to find more of those. But that's going to vary a little bit on what the read is. In the case of a futility read, that gets a little bit into ethics because you have information that may support drug doesn't work, and exposing more subjects may not align with a risk benefit on that. For stop for efficacy, 100%. You want to make sure that you're moving those subjects off the efficacy portion of the trial, because you don't want to generate a situation where if the agency does go and look at efficacy in a larger population, all of a sudden the read is a little different than what you got at your interim. So move them over to the safety follow-up and just get safety information on them.
Yeah, and maybe just more generally here, Brett, how do you manage expectations with the sites with everything being so unknown?
Yeah, it's going to sound kind of simple, but err on the side of over communication with sites. You want to set expectations early with them on what their obligations are to contribute to the interim analysis, thinking mostly about timely data entry, again, data cleaning, knowing when in advance or how planning your monitoring visits, if you're going to go on site to SDV data as far in advance as possible, and continuing to work with sites and make them feel like that they're an active partner in planning for the interim analysis. And that, again, it's not just a burden on top of needing to continue to enroll and see patients over the life cycle of the trial.
Yeah, thank you for that. Do you have to monitor SDV 100% of the data for the interim, Brett?
Yeah, so we covered this a little while ago. Again, not necessarily. As Ben said, what you really want to look at is what's your critical data that you're evaluating specifically for that interim analysis, using that as a starting point. One thing that I will say that's somewhat adjacent to that question is what your process is going to be for, and I'll loosely say locking the database, but it's really not a lock. It's more of a freeze, is the terminology that we'll often use. What we find to be most effective is what we call a rolling data freeze. And what we will do that as patients complete the time point for the interim analysis, we will review, ensure that the critical data points we've identified are clean, and at that point we will freeze those patients so that no additional changes can be made to data. We find that when we put that rolling freeze plan in place, we're able to then only have a few subjects left as we approach the actual interim analysis cutoff point that would left to be frozen. So again, it all goes back to to early and efficient planning.
I'll say to Ben, did you have any more thoughts here on this?
No, I was just going to say that we have a couple questions in the chat, and we're coming close to time. So why don't we move over to some Q&A. So if anyone has any questions that they haven't hit yet, make sure to drop those in the chat, and we'll hit the few ones that we have here already. So do you need to have a plan for the IA in the first protocol, or can you do an amendment for an IA after the trial has started? You can do an amendment. That's perfectly fine, seen it done multiple times. Just make sure that you build in time to get that agency feedback. They're not going to be on any kind of PDUFA clock, so they will get back to you when they feel like it. And so you will be creeping towards that 50% or whatever you had planned and not have any agency feedback, or you have outstanding questions in the SAP, and that can be a little nerve-wracking. So yes, it's possible, but maybe try to avoid that just because the timing cannot work out perfectly every single time. And also, you may just hit the point where you have to do it without agency input, which leaves you at a lot of risk. The other option there is also, if you do get in that scenario, you don't have to look at it right at that timing. So if you have enough overall timeline, you can, 75% of the way through, look at what it was back at 50%, and then do a decision based off of that. Is this a good idea? No, not really. There are some other operational considerations with that, because you're going to be a lot deeper in the trial when all of a sudden you're saying, hey, we need to increase based on back what we were seeing at the 50%. You could also do it at that 75%, but all of a sudden you're going to hit this moving target where what your SAP and your protocol need to say for these adjustments are not going to be locked down. So there's a lot of complexity with that. Try to get it in your initial draft or an early draft so that you do get that agency feedback and do get everything resolved. Okay.
Just to echo on that, if I can, the earlier the better. If you can't get it done ahead of time, the longer you go into the trial, the more likely the agency is to suspect maybe that you're peeking at the data and wanting to adjust accordingly. And so some sponsors will say we need to have all these documents nailed down and agreed to before we have our first site even active, or at the very least until the first patient is in. Just anything you can do to minimize concerns of impropriety.
Yeah, and I think one last point on that is this is very much buy the ticket, take the ride. Once you look at that data, you've kind of rung a bell that can't be unrung, and you need to accept what that read is and kind of follow what you've put in your charter. If you kind of deviate from that, you are putting yourself at risk where, again, the agency may determine that it's not an adequate, well-controlled trial. If you made some decision which is not spelled out in the charter, but could have been a result of the read that you got from it. Again, information is moving from this unblinded space to people who are decision makers. And if you make decisions not in the charter, FDA may view that as compromising the trial and it's no longer adequate and well-controlled. So next question I see, clinical benefit with IO therapies is often delayed. How would one mitigate against such scenarios where survival benefit is delayed? That's a place where potentially an interim analysis would not work great. What you need to do with the interim analysis is that you're identifying a cohort of people who are evaluable in a somewhat final way. And with what's described here and survival analyses in general, as long as someone is alive, you don't have final locked data on them, so to speak. And so potentially not the best approach for those types of trials. There are interim analyses that can be done there, and that's probably a separate topic, and we'd love to talk to you offline in a separate forum. Is there a standard threshold for futility, or are they all determined for each study? It's a little bit of both. Frequently, I like 20% conditional power. So that says, based on what we've seen so far, there's only a 1 in 5 chance that you can pull this out. But you can set it lower. You can set it higher. The math around where you would do a sample size increase, if you start out with like 90% power, it's somewhere down around like 40% is where the sample size increase stops making sense. So that could be used as a futility boundary where but it's a little bit too much of a dichotomy there where you're saying either we increase the sample size so we can hit it, or we just take our ball and go home. And so usually there's a little bit of a gap between the two. The other thing that you have to consider is what that is saying about your current effect size. Do you have a clinically meaningful effect, or is what you're seeing at the interim passing a threshold where you're like, maybe we could get a statistical significant outcome, but we can't market this drug because there are better things on the market. So it's pretty much study by study. You need to look at your sample size, the operating characteristics of, what does it actually mean for a given threshold?
Excellent. Well, we're going to stop there today. This has been a real treat, and I'd like to thank our speakers, Brett, Ben, and Scott, for a great discussion today. We really appreciate you sharing your insights and expertise with us, and I'd also like to thank our audience for joining us today and your great questions that came through. We hope to see you again soon. Have a great day, everyone.